Hi. Good morning, everybody. Hello, I'm Mary Beth Fitz with the North Carolina Office of State Archaeology. Welcome to our lecture series, and thank you for joining us. This year, in coordination with the Department of Natural and Cultural Resources' She Changed the World campaign, we are celebrating the centennial anniversary of the women's suffrage movement, sponsoring Trailblazers lectures by and about women in North Carolina archaeology and history. Today we are honored to welcome Dr. Nora Reber. Dr. Reber is an associate professor of archaeology in the UNC Wilmington Anthropology Department. She received her PhD in 2001 from Harvard University, where her dissertation research focused on the detection of maize in absorbed pottery residues. She is the author of numerous publications and technical reports, including an entry for the archaeological applications of gas chromatography, mass spectrometry in the Encyclopedia of Global Archaeology, and articles in Archaeometry, Advances in Archaeological Practice, the Mid-Continental Journal of Archaeology, the Journal of Archaeological Science, and American Antiquity. Dr. Reber's current research interests include archaeological pottery residues, rice plantations in the lower Cape Fear River Valley, the analysis of absorbed visible archaeological residues, absorbed and visible, form-function relationships in pottery, light stable isotope analysis, and the rise of complexity in the American bottom. Her laboratory, the UNC Wilmington Pottery Residue Lab, is a dedicated facility for gas chromatography mass spectrometry, analysis of absorbed and visible pottery residues. The lab has analyzed samples from across the Southeast and as far away as Cuba. Today, Dr. Reber will discuss her work regarding pottery from one of our state's significant historic sites in a talk entitled The Chemical Trowel, using absorbed pottery residue analysis to interpret pottery use at Brunswick Town, Fort Anderson. Please join me in welcoming Dr. Nora Reber.
Thank you, Mary Beth. Is that good? Okay. So this is kind of a twist of topic into trailblazers and women in archaeology. What I'd like us to do is focus on trowels and women in archaeology in two separate ways, I guess. Firstly, when we're talking about pottery residue analysis, people often find it a little intimidating. It sounds so complicated and magical, like you can see just what people are always eating. But in reality, residue analysis is best thought of as another archaeological tool, like a trowel, just more expensive, basically, and prone to breaking. Like most archaeological tools, its use is fairly easy. It's the interpretation that is difficult. It's easier with experience, and it always involves uncertainty, so no magical answers. And again, one of the ways that archaeological pottery residue is sort of more akin to archaeology and less to straight analytical chemistry is that taphonomy is really, and site processes are really essential in interpreting pottery residues. So that's the first way I want you to think of as trowels, as residue analysis like a tool. And secondly, one of the things I want you to remember throughout this talk is that when we're talking about pottery residues, we're probably talking about things done by women while they were using pottery, while they were processing things in the pot. So not all the time, but I think it is possible to give a gendered look at pottery use and residues when we do this analysis. Although honestly, we're often taken up so much by the technical challenges that we may not get there. So first what I'd like to do is just sort of give a little background, talk about how this chemical trowel works, and then we'll get into sort of the cases, the case study of different residues from pottery sherds excavated at Brunswick Town in the William Peace College Field School from 2009. And then we'll just sort of see how it applies. So firstly, residues tend to come in two basic flavors. You get your visible residues, that's the blackened incrustation right there that everyone looks at it and they're like, wow, that's a residue. But what I tend to work with more often is an absorbed residue. So absorbed residues are residues that absorb within the actual ceramic matrix of the pot. You usually don't know they're there. So you don't usually dig up a pot with an absorbed residue and say, wow, that's it. Instead, what you do is you take a collection of pottery that seem kind of interesting to you, and then you send it to a lab, and we tell you if you have a residue or not. So the key things to know about while we look at this is firstly, pots aren't only used to cook food, right? And furthermore, you can cook food without pottery, right? So pottery residue analysis doesn't reflect the total diet of any group of people. It reflects how they use their pottery. So you'll never get things that are just roasted over the fire. You'll never get anything like pancakes, sadly. Well, not usually. You're not going to get, anything that they roasted in the embers or anything like that. You get strictly soups, stews, and maybe frying. It's sort of unfortunate, but there you go. You simply can't get a pottery residue off something that's not a piece of pottery. The other thing is that pottery can obviously be used for lots of things besides food. So processing pine resins, dyes, medicine, so on and so forth. So that means that when we're writing a residue report, we tend to be very cautious not to say the diet at the site or the food at the site. Instead, we say the resources processed in the vessel, which sounds horrible, but it does reflect the fact that we're not always talking about food. So when we're doing pottery residue analysis, people often want to know particular resources. So they say, were they eating cod or were they eating catfish? Unfortunately, for reasons that I'm about to explain, I probably can't tell you that. I can tell you total pot use, particularly with absorbed pottery residues. So in order to think about this, it really helps if we think about residue buildup in pots as being an awful lot like a cast iron frying pan. So imagine your cast iron frying pan at home, right? You just got it home from the store. The first thing you do is you season it, right? You rub it all over with oil, and you put it in the oven. What's happening is that oil is soaking into the pores of that cast iron and making it nonstick. Now, sometimes in ancient times, people did that with their unglazed pottery as well. They might have rubbed it with pine resin. They might have cooked something in it, like something sticky, like corn or just a stew to let sort of fats and oils get into the pot and make it non-sticky. But then when you have your cast iron frying pan, every time you use it, just a few of those sort of fats from the things you cooked in the pot soak into the pores of the pot, right? So the longer you have the cast iron frying pan, the better it gets, right? Unless you do something like put a lot of tomato in it and strip it out. Luckily for us, no tomatoes in prehistoric North America, so that's nice. So over time, little bits of fats from all the things you ever cooked in that cast iron frying pan are soaking right into the pores of the cast iron. And the exact same thing was happening to ancient unglazed pottery when people cooked them. So when we look at residues from a pot, particularly an absorbed residue, what we're getting is little bits of everything ever cooked in that pot over what we call its use lifetime. So sometimes, if the pot wasn't used very long, you might get quite a short time period of use. Sometimes, if the pot survived a long time, you could get multiple years of use, all sort of mishmashed together. So particularly in parts of the world where you don't have special use pottery vessels, which I'm afraid includes an awful lot of the southeastern and midwestern United States, most of the time your residue is going to come out mixed. You very seldom get just a pure, beautiful, one-shot residue. That said, I'm going to show you one today, but it was super rare. It wasn't from prehistoric times, it was European. So in cultures such as most Native American cultures and probably most slave cultures, every residue you get is going to be a mixture of components. So then the question is, which components? How much of what? And how precise can we be? And I'll talk about how we do that in a minute, but I just want to take a minute to note that it's really important to say that pottery use is cultural. What people use pots for varies from culture to culture. So what we're really looking at is cultural behavior. All right, so let's just talk about residue formation to sort of pound home the idea of how much gets into a pot and how a residue forms, and taphonomic processes. So we have this pot. It's being used to process lots and lots of things. Some of them are food, some of them are not food. Over time, it gets into, if it's a stew, like the cooking liquid, and it soaks into the wall of the pots. Now, in order to get into that cooking liquid, it's going to have to dissolve in water, right? Right. So that means you're not going to get resources processed to soak into the wall of the pot if they don't absorb in water or they don't dissolve in water. Unless you're talking about something like deep fat frying, in which case you'll get some resources, but they'll tend to be marked by high temperature use. So over time, eventually the pot will break after, a week or a year or 5 years or 10 years. And this is where this sort of taphonomic archaeological aspect of residues comes in. So we have all those bits and pieces of all those compounds that absorbed into the pot that are soluble in water, right? Well, what if you're in the southeast and it rains? Well, some things will wash out, won't they? The things that are soluble in cold water will, generally speaking, wash out. So most of what we find in absorbed residues are things that are soluble in hot water, but not cold water. That limits us essentially, not completely to lipids, but mostly lipids. Lipids and some alkaloids, like nicotine and scopolamine that everyone loves so much, and caffeine. But generally speaking, it's all lipids, so fats. And that sounds very limiting, except lipids are this huge, varied, informative class of compounds that actually tell us a lot of information. But just be aware that anything that dissolves in cold water is basically out of there after 2 rainstorms. And then your pot is buried. It's been buried in the ground. And through time, there's all this dirt around it, right? And there's water, and there's maybe some oxygen, as there is in dirt, and there's probably bacteria, right? So all those things will act on your residue and essentially change the components. So you get oxidation. That'll essentially break fats apart. You get hydrolysis. That's from the water. That also breaks fats apart. And then you get microbial breakdown. So this dreaded little bacteria will just eat your residue. So the question is, why don't they eat all of your residue? Well, this is because your residue is protected in both absorbed and visible residues by one of 2 separate things. In an absorbed residue, because the residue's absorbed within the walls of the pot, the residues or the compounds are partially protected by all that ceramic around it. They're basically soaked into the pores, and the pores are quite small. Small enough that not too many bacteria can get in there, not too much air. Water will still get in there because it's unglazed pottery. In the case of a visible residue, there's obviously no ceramic around it, but there is charcoal. And carbon, as you all probably know, is a pretty good protector of compounds. It essentially sort of protects the compounds from bacteria, not so much from oxygen, but certainly from bacteria and a little bit from water, although not so much, really. So, and then furthermore, when people dig up this pot, things can happen, right? So someone might have, they should have sunscreen on when they dig, right? Well, what happens when they pick the pot up in the screen, or the pot shirt up in the screen? A tiny little bit of sunscreen from their hands and DEET from their bug spray will almost certainly sneak into that residue. Because sunscreen and bug spray are, firstly, generally speaking, lipids, and secondly, specifically designed to absorb in things. It's what they do, right? Chemically, they're specifically designed to absorb in human skin, which has pores in it, and so it absorbs in ceramic as well. Furthermore, people don't just carry the pot shirts home on a little platter, generally speaking. They put them in bags, right? Often plastic bags. Plasticizers are notoriously labile compounds. Quite a few of those plasticizers will sneak into your residue. And then we have other things. So normally speaking, if the excavator knows they're going to submit their sherds for residue analysis, they will not, for example, wash the sherd nor label it nor anything like that, and they shouldn't. But periodically, people won't know they're interested in residue analysis until after they've washed the sherd and after they've labeled it, and then we get to deal with compounds from your nail polish and your ink and possibly your whiteout and all that kind of thing, and glue. Yeah, don't talk to me about glue. So in essence, whenever we're looking at absorbed pottery residues, it's a little like an archaeological feature to some extent. It was caused by human behavior. It changed while it was in the dirt due to the bacteria and the oxidation and the hydrolysis and so forth. And then there were a few more sort of processes that happen post-excavation, modern contaminants. And so when we get a potsherd in our lab, we essentially, so in the bottom right-hand corner there is one of my students grinding up a potsherd. So in the case of absorbed residues, I'm afraid we take some of the sherd and we powder it up in a mortar and pestle. So this is a pretty spectacularly destructive analysis. At its most, it doesn't need more than like 10 to 12 grams of sherd, and 2 is actually fine most of the time, so it's like not so much. But we powder it up. Well, first we clean the sherd very carefully with a Dremel tool to get any labeling, hopefully any bug spray and sunscreen and plasticizer off there, or as much as we can, basically. Then we grind it up. Then we extract it with a strong solvent, and the idea is that once we've mechanically destroyed the sherd, and once we put that solvent in there, that lipids in particular are very soluble in, the steric protection of the ceramic sherd will be done with, because we just destroyed it with the mortar and pestle, and then everything will be able to dissolve within the solvent. Then we just clean the solvent out, reduce it down to a very small amount, and we put it in our gas chromatograph mass spectrometer, which is that instrument you see in the top left. So there are many different instruments that can be used, obviously, to analyze chemicals. I tend to use the GC mass spec, or gas chromatograph mass spectrometer. And the reason I do this is because a GCMS is essentially a separation machine. So the first thing it does is it takes complex mixtures and it separates them out. So you've all done this experiment when you were kids with like the ink and the blotting paper and the different colors in the ink came up in the water. That was a chromatographic separation. So different of the constituent parts of the inks traveled at different speeds, and that's why it's separated. That's exactly how gas chromatography works, except you're running gas through instead of water. And instead of blotting paper, you have anywhere between 15 and 30 meters of something that looks a lot like fiber optic cable. Very, very thin, narrow columns. So with all that length and with all that narrowness, many of the complex compounds in your mixtures will basically get separated out. Then you can attach practically whatever detector you want to the end of it, and I like a mass spectrometer for this. So the GC separates it, and the reason I like a mass spectrometer is it essentially tells you what's in it, or it tries anyway. So this picture up here is a picture of a complex mixture that's been separated. This is actually a yaupon tea that we processed and then in a vessel, and then we buried the vessel in my backyard for 6 months. So you see this particular residue went through cooking, and then it went even through some site processes. And you can see there's still a fair amount of stuff in there, including that thick, nasty-looking peak there. That's caffeine. So you can see that it's surviving quite well. Of course, it's only been 6 months, but you can only do so much with keeping your experimental work really accurate before you die, before it's done with. So we see how all those complex mixtures separates. And what the mass spectrometer does is it's essentially like a huge, immense light bulb filament, at least mine is. And it zaps everything as it comes out the end of the column, it zaps it, and the current from the filament breaks the molecule apart at its weak points. Since every molecule has different weak points, you essentially get what's like a fingerprint of all the different weights of the molecule fragments, and then these can be interpreted. Now, everyone always kind of hopes, particularly when they start this type of analysis, that you can run this stuff through the system and then you can press identify and it'll tell you exactly what's in there and it'll be like magic. And with the common compounds, this works and it's like magic. The problem with residue analysis is that we tend to have a complicated mixture, as you saw from our previous slide, and some of these compounds are quite obscure. I was reading an article the other day that said that, millions and probably billions of chemical compounds are known in the organic world, but the largest database used in mass spectrometry is 800,000. So you're not always going to get a perfect match. That means me, the analyst, gets to try to reconstruct the unknowns by essentially working backwards from the molecular weaknesses. Sometimes this is easier than other times. And this is one of the reasons that I said that this kind of work becomes easier with experience. Over the years, you see more and more stuff, and it becomes easier to identify. So just for your information, this particular MS, or mass spectra, is of a very common fatty acid, palmitic acid, or I usually call it C16O. So it's a nice, plain, straight, unsaturated fatty acid. If you all ate breakfast, which is good for you, everything you ate had palmitic acid in it, pretty much. Yeah, unless you had black coffee. It's in everything. So it's very common. It always gets identified. and we'll see it again quite soon. So where the interpretation comes in and where it can become difficult is trying to work backwards, given that you know your residue probably came from multiple different resources, right? So you have to try to figure out or identify what sorts of resources you've got. And this is where we get into what's called the biomarker approach. So biomarkers are essentially compounds that are unique to a particular resource or class of resources. So usually speaking, you get what I call category biomarkers. So like cholesterol, this is one of the more common ones. Tells you it's probably either meat, fish, or shellfish, right? So some sort of a critter or animal. And I'm defining shellfish as critters for, the reasons of residue analysis. You also get plant sterol, sitosterol, camposterol, stigmasterol. These are biomarkers for plants. So if you get one of those, you know you have plants. That's nice. You also have things like, for example, dehydroabiatic acid. It's a common oxidative byproduct of, I'm going to sound super technical for a minute, but bear with me, abiatane and pimarine resins. And this is telling us it's pine, basically, or to be precise, it's telling us it's a conifer resin, probably from pine, but also possibly from members of family Cupressaceae, which for the purposes of North American archaeology includes all the cedars and cypresses. And since dehydroabiatic acid, I call it DHA for short, is a breakdown product, it's super stable, and a lot of other compounds turned into it. Since it's so stable, if it's there, it doesn't wash out. Guess I don't have it in a slide here. You also get, and if we're really lucky, we'll get them resource-specific biomarkers. So caffeine, obviously, more than one thing has caffeine, as you all know, tea, coffee, chocolate, and so on. But each of these resources comes from different geographic areas, doesn't it? So before you're in the modern world, if you find caffeine in the southeast, you're probably looking at yaupon or black drink. If you find it in Mexico, probably chocolate. If you find it in Africa, probably coffee. If you find it in China, probably tea. If you find it in Brazil, probably mate. So there are multiple sources of them, but they're all geographically limited. So that's nice. Now, if you've got some trade situations, areas that are not quite in the heartland of any of these resources, then you're looking at other evidence, as archaeologists so often do. But in essence, caffeine is resource-specific. Betulin is resource-specific for birch trees, should you have any birch trees. Sadly, since I mostly work in the southeast and Midwest, I don't find it very often, but we can hope. And you also get what are called process biomarkers. So these are compounds that weren't in the original resource, but they formed during the cooking process. We'll see some of those today. So for example, if you fry fish or highly unsaturated fatty acids at high temperature, you get this cool, super stable fatty acid derivative called an alkylphenic alkanoic acid. So remember when people were frying things in McDonald's French fry oil and people were saying that toxic chemicals formed in it when the oil got old? That's them. They may be toxic, but they're really stable in a residue. So if you find this, people are cooking very unsaturated, resources with very unsaturated fatty acids at very high temperatures. So 9 times out of 10, it's fish, because fish and shellfish have very unsaturated fatty acids. Not required by natural law or anything, but it's usually fish or shellfish. Pyrolytic ketones are what happens if fatty acids are heated at temperatures between 300 and 350 degrees Celsius, not Fahrenheit. So pretty hot. And so that tells you essentially that someone was cooking something with fatty acids, which includes everything, but at this very high temperature. Hotter than you'll ever get while making a stew, because stew, of course, tops out at about 100 C, or maybe a little more, but not much. Everyone with me so far? Okay. So let's talk about what you do if you haven't got a biomarker, or if you have some biomarkers, but you want further interpretations. Here we have to get into fatty acids, and this is difficult. Fatty acids are, like I said, the most common lipid in any resource, pretty much ever. There are certain guidelines, we might say, about them. Plant fatty acids are more unsaturated. That means they have more double bonds in them than meat resources. Fish and shellfish fatty acids are even more unsaturated, usually having between 2 and 5 double bonds in some of the more common fatty acids. So you would think that all we would have to do would be to look at a residue, match up the fatty acid ratios, do some math, and we'd be there, right? We'd know. But I said that most residues are mixed, right? So what happens if someone's cooking like deer meat and fish? It averages out to a plant, pretty much. Then we have some other problems, and this goes to these taphonomic issues that I mentioned. So double bonds are more sensitive to hydrolysis and oxidation than single bonds. That's why like fish stinks after 2 days. That'll be all the unsaturations in your fatty acids oxidizing on you. And this happens to residues as well. So the very unsaturated fatty acids that are common in fish and shellfish will break down much, much, much more rapidly than the less saturated and unsaturated compounds. So as a result, a very unsaturated fish or shellfish residue will, give it 50 to 100 years. It's going to look a lot like a plant, almost identical to a plant, in fact. And furthermore, long fatty acids tend to break down more slowly than short ones or wash out less. So that means that within if you want to call it the residue record, there's a bias towards long unsaturated lipids and a bias against short unsaturated ones. This is why identifying fish and shellfish is always very tricky. And also why sort of in many cases the default interpretation tends to be something quite like could be meat, because give something enough time, everything looks like meat, pretty much. That said, this residue here this isn't for one of the ones I'm talking about today, but this is a fatty acid extract in my methods from a very unsaturated compound, probably a fish or shellfish, as I glance at it. Oh, I don't have a laser pointer. Oh, wait. No, I don't. We see there are a lot of very long fatty acids. There's an unusual amount of unsaturations in some of them. So where it says 18-1, that means it's 18 carbons long with one double bond to it. So in essence, what we do with our fatty acids is we kind of wave our hands around in an irritatingly unscientific and yet archaeological way and say, it's probably plant or fish, shellfish, or it was a mixture of meat and plants, but we think it was mostly plants because of this degree of unsaturation. So there's not really a bright line. I mean, everyone wants there to be, but owing to the mixing and the taphonomic process issues, that's usually not possible. Okay, so deep breath. I guess there's going to be a Q&A session at the end. Otherwise, I'd sort of ask for questions now, but hold them, I guess. And so now we'll talk about some of the specific applications of this technique. To the Peace College Field School of 2009, where they collected a bunch of sherds from this barracks west of Battery A at Fort Anderson. So they actually collected a whole bunch of sherds, and it's worth noting that most of the preliminary work on this was done by Matt Kerr, who at the time was an undergraduate student of ours, and right now he's at University of Tennessee, Knoxville, getting his PhD. So most of the initial work on this was all Matt. But of the larger collection of sherds collected, we analyzed 9 prehistoric sherds, 3 colonaware and 2 historic unglazed earthenware, probably like an olive jar or an oil jar, and one thing they called the mystery sherd, which was sort of like a blob of dirt that we think was probably some sort of a pot sherd, but it's hard to tell. So what I'm going to do, and this is kind of a small sample size, but it's what we've got. One of the things we try to work towards a lot in residue analysis is larger sample sizes. And this is because pottery residue analysis is, like I said, a lot like archaeology. And the more you dig, the more you learn, and the same goes with residues. So although usually we're doing these small proof of concept things, like this was, I think, 14 sherds that were easily identifiable, it's a little like trying to interpret a site from digging up 14 features. You can do it. It's not going to be very specific. So more is, generally speaking, better. But what I'm going to do is go through this relatively small group of samples in chronological order, starting with prehistoric, which was our largest sample size, 9, then going into our colonaware, and then I'll compare that with some other colonaware sherds I looked at from the Dean Hall site in South Carolina, thanks to Brockington and Associates. And then I'll move on to our 2 historic unglazed earthenware sherds, and then I expect we'll have run out of time. So of our 9 prehistoric sherds, just some quick chronological information. 7 were from levels interpreted as colonial, and 2 are from undisturbed prehistoric. Now, whether those 7 sherds really came from the colonial period or whether they represented disturbance of the earlier prehistoric levels is really hard to tell. And thanks to Tom Beeman for his long and detailed report, which I used a lot while working on this talk. So looking at this, there were 3 net-impressed sherds, 3 cord-marked sherds, and 3 fabric-impressed sherds. So it was nicely spread across the different types. As we look at interpretations, we had probable fish and shellfish in 5 of those 9 sherds. So that's kind of a lot, but not surprising given where we are, right? I mean, Barracks West of Battery A is right next to the Brunswick River. So we would expect some preponderance of fish, shellfish. Now, you may ask how I interpreted fish and shellfish, given that I just said it was really, really difficult. The fatty acids from these 5 sherds were very unsaturated. And when I say very unsaturated, I mean we were finding the fatty acid arachidonic acid, or 20:4. So that's 4 double bonds and 20 carbons, very typical of fish and shellfish. Preservation on it is not usually good, so the fact that we were finding it kind of definitely tells us there was a lot there to begin with. Further, there's sort of a secondary method for fish identification developed by Baton et al., which was developed for North Sea fish in Denmark, but it works pretty much for all fish because the lipids are similar. What you do is you look for highly, highly unsaturated fatty acids with just a lot of palmitic acid and a very tiny amount of the C18 stearic acid. And then you look also for the presence of cholesterol, which is super abundant in fish and shellfish, nearly all of them. If all those 3 requirements are sort of fulfilled, you can reckon it's probably fish or shellfish. It's not a guarantee. That said, these 5 sherds pretty much all fit those requirements. They were highly unsaturated with this C24. They had, in some cases, just sort of hysterically minuscule amounts of C18O and quite large amounts of C16O, and they all had cholesterol in it. So I'm interpreting them as primarily fish and shellfish, but they all had plants in it because they were plant sterols in there. That sitosterol and camphosterol that I mentioned earlier. And I'm just dreaded. I thought I had a slide here. Sorry about that. Yeah. Then we had a mixture of plants and meat with plants predominating in 3 of those residues. And interestingly enough, that was from 1 cord-marked, 1 net-impressed, and 1 fabric-impressed, so it spread like sort of elegantly across our 3 types. And of our fish and shellfish sherds, 2 of them were net-impressed, 2 were fabric-impressed, and 1 was cord-marked. So we're not seeing much differentiation between decoration and pottery use, at least from this small sample size. And then we had 1 sort of unusual sherd that was strictly plants, no cholesterol, highly unsaturated, but it did have the sterile biomarker for the presence of fungus. That's ergosterol. Now, when we're interpreting fungus, you have to be really careful because ergosterol is a category biomarker for fungus, but fungi is a really, really large family of things, right? It's everywhere between edible mushrooms, like that, to just the fungus that grows on your residue after people bury it. So usually a little bit of ergosterol, we just reckon the residue grew a fungus. But there was a lot of ergosterol in these sherds, in some cases very, very large. So in 3 of the sherds, there was so much ergosterol that I tentatively think there may have been some mushrooms going on in there, or something like that. So, I mean, further work will need to be done to find out what sort of edible fungus might be in that area. But yeah, there was just so much ergosterol. I think that's the most likely interpretation. The other possibility is that these sherds essentially rotted on the surface for a very, very long time before they were buried, which I suppose is possible, but seems a little unlikely to me. Of the other 3 sherds that had some ergosterol in it, it wasn't out of the ordinary. I think they just rotted out a little bit. Like I say, it happens. Residues do break down. So what we may be looking at in some of these cases is a high incidence of fish, shellfish, and perhaps some mushrooms cooked with that. But again, I can never give people the magic answer they want. And then we had one of these sherds interpreted as a mixture of plant and meat, but it had sort of some unusual compounds in there. So it had these things called hopanes. Hopanes are usually, well, they show up a lot in bacteria, but as a practical matter, they show up in very old bacteria, and they're usually a biomarker for petroleum oil. Sometimes they show up and we figure it comes from the soil, or it's just an accident, or maybe someone dropped some gas on them at some point in their use lifetime. But I'm mentioning this because we're going to find more hopanes at this site, and also, in part probably because Matt was involved with taking samples in this project, we got soil samples from this site, and there's no oil in the soil. So if it is contamination, it wasn't from the soil around some of these sherds. So that's interesting. And as we look at this, I just want to show you this to show you first like what hopane looks like when it's present in very small amounts, and also to see sort of what we're working with. So that first figure up there with a really tall thin peak, that's what you get when you run things through the GC mass spec. The tall peak is our internal standard. We add that to quantify things and just make sure everything's working. But you see that all those peaks from the actual residue are very, very tiny compared to our internal standard. You often don't get boatloads of residue. Sometimes you do, but not always. So down here is actually our inset showing like what happens when you blow all these things up. And you see, although it looks small, there's actually a lot of things going on in there. We have the whole series of alkanol suggesting plant waxes. We have cholesterol telling us meat or fish, shellfish. We have sitosterol telling us plants, and we have that two weird little hopanes. Hopane and hopine. But they're not overwhelming at all. Good so far? So take-home message on our prehistoric sherds is a stew, a mixture like we usually get, but a lot of fish, shellfish, a lot of plants, meat also present, possibly fungus, and no variation across the different vessel types. So then we go into our colonaware. Since we only had 3, I'm going to go through them in a little more detail. And these were very interesting residues, even though the sample size is really too small to make any large, dramatic, awesome claims. So this slide here shows us what we call the total lipid extract of one of these colonaware sherds. This will be sample 2. And it has a whole bunch of triacylglycerols in it. This is a really well preserved residue. It came out very, very well. So of these 3 samples, all of them had a mixture of fish or shellfish and meat, but there were some particularities. One was primarily fish or shellfish, based on the fatty acids, but then it also had conifer resin, not too surprising given that it's setting right in a longleaf pine area, which is known during the colonaware period to be a major sort of center for naval stores production. And then also some hopanes. How odd. Then we have a second sample that shows some of the or possibly uses a lamp, but I'm thinking, because it was a cooking vessel, more high temperature cooking, such as frying. Sort of a shallow fat frying will give you this exact effect. We get a mixture of plant and meat resources in this. And that's actually the figure I'm showing you here. And then our final colonaware residue was primarily plant with a mixture of either meat or fish, shellfish, couldn't tell, and conifer was present. So this already looks quite different from our prehistoric sherds or our prehistoric residues. None of the prehistoric residues had any evidence for conifer resin, but the colonaware did in 2 out of 3. And in the first one we see the biomarker retine. It's an unusual process biomarker. It's probably telling us pine tar or pine pitch. It's what happens if you cook conifer resin hot and long, such as you do when you're making pine tar or pine pitch. So we're seeing this use of conifer resin and deep fat frying, which is different from what we saw in the prehistoric period. Since it's only 3 sherds, and since there's not a lot you can do with 3 sherds, I thought I'd compare these with the 8 colonaware sherds I did from the Dean Hall site in South Carolina. So this is a rice plantation, 18th century, a lot of colonaware, much more than was ever found at Brunswick Town, Fort Anderson. And as you can see from this picture, there are some pretty cool colonaware vessels in there. So of the 8 samples, one of them appeared to have been empty, no residue in it. So that means it was either never used at all, broke right out of the kiln or right out of the firing. It may have been used to process things like water, like to hold water, or to hold dry ingredients. Dry things don't absorb into residues. Can't. There's no substrate to move it over. Or it may potentially have been used for serving. Very hard to tell. 5 of the sherds showed a mixture of plant and meat, sort of our old friend the stew. 1 of them showed probable fish and shellfish plus pine pitch, and conifer, and as well plants. So a residue that looked quite similar to some of the ones that we're seeing at the Brunswick Town Fort Anderson. In none of these sherds do we see any hopanes, so no petroleum oil, nothing like that. We do see a higher incidence of, well, probably a similar incidence of conifer resins. And then we come to our historic unglazed potsherd. So you know how I said, oh, here's my laser pointer. You know how I said you normally don't get a single-use vessel? Yeah, it's a single-use vessel. So, and this also sort of speaks to what I was talking about earlier. When we originally did this analysis, which we did in 2009, I had no idea what was in this sherd. So this is actually a sherd I did later that clued me in. And this is the one I did in 2009 from Brunswick Town. And we saw that huge, nasty-looking lump on there. And all we knew was that it was a huge, nasty-looking lump, which indicated serious polymerization of all of our compounds. We couldn't figure out what it was. But then, several years later, I was working on a project with the University of Louisiana at Lafayette on the effect of the Deepwater Horizon oil spill on pottery residues. And I saw that. Look, it's the same big lump. It turns out to be called the unresolved complex mixture, which is a scientific term for what oil looks like in a residue because it's such a mess. But if one looks at it more carefully, we can find these biomarkers for petroleum oil, like hopanes. See, here they are. And steranes. So I went back and I looked, and there they were. Our historic earthenware vessel was holding some sort of petroleum oil derivative. That appears to be, I can't say it was all petroleum oil derivative, but it was so much that it overwhelmed anything else that might have been in there. So that was interesting. Of the 2 unglazed sherds we did, I think they were probably from the same vessel, probably some sort of a Spanish olive jar, Spanish oil jar thing. The residues were both the same unresolved complex mixture, kind of a mess. So I'm thinking they're both from the same vessel, which was used to carry something oil-ish. Could have been tar, could have been crude oil, though I don't know why they would have. Tar seems more likely, bitumen, anything like that. But from a residue standpoint, it looks quite like crude oil. And I just wanted to sort of show some of the ways we can pull apart, I suppose, sort of trowel back some of the complexities of these. So this was my other unglazed earthenware sherd, and you can see it's just a straight lumpy mess. It's worse than my other one. What you can do is with the mass spectrometer, you can pull out just the molecular weights you're interested in. So in this case, I just pulled out 191, which is an important peak in hopanes. And you can see that we're actually getting a whole bunch of hopanes in there. And if you do the same thing for 217, which is a crucial sterane mass weight, you can do the same thing. And so that allows me to see that even though it's sort of an unresolved lump, that it's definitely derived from petroleum oil. Now, one of the interesting things about petroleum oil residues that I learned about while working on the Deepwater Horizon thing is you can actually sometimes geographically identify crude oils by looking at the ratios of hopanes to steranes and matching them up with oil basins around the world. The problem is that all these ratios are a little bit sensitive to stuff like heat and, unfortunately, bacterial breakdown. Plus, there are a lot of different oil basins in the world, so I haven't been able to do this sort of analysis yet on these sherds. It might be possible. Or it might all have been too transformed by whatever happened to the residue. It doesn't work as well with residue analysis as it does if you have a lovely fresh crude oil sample, which is what it's usually done for. So then we have to ask ourselves, well, why does someone have what appears to be an oil jar full of tar at Brunswick Town, which is well known to be a major capital of pine tar production? So why would they need non-pine tar? And that I still don't know the answer for. This unglazed earthenware vessel was associated with a colonial structure, a high-status colonial structure. Looking just very quickly at some of the uses for oil and oil derivatives during the 1700s, sometimes it was a medicine. Could have been. It was also used for brown paint, but that seems unlikely to me. Interestingly enough, in the article by John Mintz and Tom Beeman on oil jars from Brunswicktown, I found a quick mention of the fact that tar was sometimes found shipped in Spanish oil vessels. So I don't know what they were using it for, but we can hope. And if so, I think that's what we have here. We have a Spanish oil or olive jar containing tar for unknown purposes. And I think as we look at this, this tells us the way that cultural use of pottery has changed through time at this site. In the prehistoric period, we've got these mixed residues showing fish, shellfish, plants, meat, maybe fungus. No pine residue, which is a little interesting. The colonaware showed pine tar, pine pitch, a new cooking technique, frying, and a mixed diet, including fish or shellfish, but I would say not that much different from the prehistoric Indians in terms of the dependence on fish and shellfish. And then when we get to historic, we're seeing straight single-use, pots that are used for one thing, shipping tar. Now, it may have been recycled later, but if so, this particular vessel was so full up of petroleum residue that we just can't see it. It was sort of all overcome by the oil residue. So we're seeing changing cultural use of this pottery, and we're seeing as well differences in who is using the pottery. So we can reckon that our prehistoric cooking was probably done by women most of the time. We can figure our colonaware pottery was probably used by women most of the time. But then we can figure our historic unglazed sherd was probably loaded and unloaded by men as it's industrially moved in and out of the port at Brunswicktown. And that leaves us with, as usual in residue analysis, more questions than we started off with. Would colonaware results look different if we had more sherds to work with? Why are we getting hopanes in prehistoric as well as historic? That's weird. Maybe there was some washing over from one area to another, but then it should have been in the soil. And what on earth are they doing with all that tar at Brunswicktown? And this usually happens again with residue analysis. You start out thinking you're going to answer all these questions, and you answer some of them, but you come away with even more questions, new ones. And in that way, residue analysis is, again, very, very like archaeology. I'd just like to thank all the people whose work I sort of shamelessly piggybacked on here. So it was all Matt's original sample collection and residue analysis. Tom Beeman and Vinnie Malomo did the original excavations. Brockington and Associates, Andrew Aga and Nicole Eisenbarger got us the Dean Hall samples. And of course, Jim McKee presides over Brunswicktown Fort Anderson. Thank you very much, and I guess we're ready for questions.
Thank you for that talk. I'm curious, are you surprised by the combination of the pine resins with more of a food base, or could that have been like as you mentioned before, a way to provide a waterproofing or maybe a seasoning to the pots?
Yeah, actually, no. One of the things I've found more and more often as I work particularly with North American residues is they have conifer resins in them practically all the time. The thing that surprised me was the prehistoric ones didn't have any pine. They almost should have. I've done a lot of projects from a lot of different places. I just finished one from Ohio that was chock full of conifer resins. I just did one from Kansas, chock full of conifer resins, despite the fact that there are no native pine trees in Kansas. So that was puzzling. I think they were probably from red cedar. We find pine resins or conifer resins in New York. We find them in North Carolina, but that's not surprising. They appear to have just been very commonly used either as a sealant, as a flavoring. And I actually have a recent paper out in archaeometry with several colleagues with Matt Kerr and with several colleagues from Bristol that suggests that if you fire pottery over enough pine wood, you're simultaneously, in a way, sealing them with soot, and you'll get pine resin in it when you do the residues, which might answer some of our questions. So yeah, honestly, I'm never surprised when we find evidence of conifer resin anymore. I'm almost surprised when we don't find it, and particularly when we look at gullah culture and we look at, the naval stores industry at Brunswick Town. Yeah, I would expect pine in some flavor or another, turpentine as a medicine, pine wood and like soot almost flying around in the air. I mean, the air quality couldn't have been good during the naval stores period, I think. So yeah, I wasn't surprised. The Hopanes, those were weird.
I'm curious if there are any environments like coastal environments, Piedmont, that are better or worse for residues, or any geographic regions that you sort of can or generally can't get residue analysis from?
Yeah, that's a really good question. You can get residue analysis from practically any environment, and we sort of prove it from these Brunswick Town Fort Anderson residues. Because what does affect residue preservation is essentially particle size, rainfall, and salinization, with sandier being worse, rainfall being worse, and salt in the soil being worse, which means that the Brunswick Town Fort Anderson area is like a trifecta of lousy preservation for residues. Now, of course, the historic residues are more recent, which is probably why the Kelowna residue looked just so pretty. Like, that was very nice. The prehistoric ones, not so much, actually. But I did some experimental work on this in graduate school, actually, and it seems likely that clay, possibly because of the small particle size, is better for residue preservation. And of course, all organic preservation is better if there aren't as many state changes. So if it's okay if it's wet, as long as it stays waterlogged forever. It's okay if it's dry, as long as it stays dry forever. Same with frozen. But most of the Southeast is like the anti of all of that. It's always wet, dry. It's always hot, cold. So it's very bad for organics under most circumstances. Residues are certainly protected more than, cloth or fiber or anything like that by its ceramic, but it is affected. And I think you do find a higher bacterial load often in some of these southeastern sites than in, I've worked some in New York. Not so much with the bacteria up there. I mean, it's still there, but not as much. And in fact, I think when we do experimental work here, which, as I showed you in that one slide, we still periodically do, I always bury things in my backyard because it's there, but we also usually send some stuff up here to the Piedmont or somewhere, because Wilmington soil or my backyard soil is just so sandy, and it's so hot, and it's so wet. You really want a more normal clayey soil to act as comparison for experimental purposes, in my view.
Do you ever find that certain glaze compounds, I'm thinking, for example, tin glazed or lead glazed earthenware, can interfere and impede the absorption of residue?
Well, yes, and this is why most residue analysis is never done on glazed pottery. So the reason I could do that historic earthenware, it was an unglazed earthenware, kind of a oil jar kind of a thing. The whole absorption process is completely hindered, destroyed by the presence of a glaze. I tried once to get residues off glass vessels. We thought it should be worth a swing. You could kind of see residues in there, but preservation wasn't good enough. It appears, unless it's a really thick, juicy, lovely, visible residue, which is not so common, absorbed residues can really only be done on unglazed pottery, whether it be from historic, prehistoric, protohistoric, but yeah, glazes won't do it for you.
Is the residue absorption rate supposed to be covering the entire sherd, or is it possible that certain sherds you'll have high spots on just the sherd? I mean, is it possible for a double-blind study to potentially differentiate whether it covers the entire sherd or just a little bit of the sherd?
Yeah, this is a good question. So there's actually two things going on here. There was early experimental work done by charters in which you looked at where residue absorbed most within any one sherd. And the answer depends, unsurprisingly but annoyingly, it depends what your sherd was used for. If it's a cooking sherd, the lipids are usually going to go up to the top, like if you imagine you're making a soup, you know how all the like fat and stuff floats up to the top. So in unglazed pottery terms, that means the best place to sample a cooking pot is usually the neck. Or if you can see a boiling line, right on the boiling line. Now, if it's a serving vessel, then all the lipids are like going to soak down to the bottom, and you should sample the bottom. You shouldn't do that in a cooking vessel, because in most cooking vessels, the bottom of the pot is closest to the heat, right? And the heat is doing all kinds of unpleasant things to the compounds and like causing them to react and burn out and things, which is why you want that neck area. So for cooking vessels, you want to sample the neck. For serving vessels, you want the base of the pot. And she had a whole series of other ones. Stephanie Charters worked in England, so she was working on Anglo-Saxon unglazed pottery. So she looked at the whole range of different special use Anglo-Saxon pots. But in essence, what you do is you try to figure what your pot was used for and then figure where the lipid was. In terms of the Northeast, or in terms of the Southeast and the Midwest, usually I tell people to go with the neck, because most of the time it's cooking, probably. And if you're not sure, well, it's still a nice spot. Or if you can find a boiling point place, that's always the best location. Now, the other issue, actually, charters argued that if you carefully sampled different parts of a pot, you could reverse engineer it and figure out what the pot was used for. That was actually what she was going for. But it turns out to be so time-consuming and expensive to do all those samples that no one's ever actually I did it once. But no one else besides me and Stephanie have ever done it. Because for one thing, you have to have a whole pot, and then you have to have an excavator who doesn't mind you hacking out multiple little samples. And then you have to have the time to go in and do it all. So it's doable, but given our sampling issues, it's probably a better use of your time and money to take a wide spread, 5 different pots instead of 5 samples from the one. Now, the other issue there is, could a pot ever get full of residue? People have also done studies where they look at where the lipid absorbs into the pot, and unsurprisingly, it absorbs on the surface where the food is, right? So on the interior surface. And so the idea is it sort of gets soaked into the layers of the pot through time, and so you find most of the residue right on that interior surface, or just maybe a little bit behind it. And there isn't so much on the exterior surface. Now, from a sampling standpoint, this is useful because if you have someone who doesn't want to destroy the whole pot, you can go in there very carefully with a Dremel and just Dremel out the inside of the pot and, go from there. Now, where that gets difficult is we also like to clean the all the surfaces of our pot to get rid of contamination. So you must be very careful when you do this. When you clean your sherd, you have to get enough off to get your finger touches and your plastic bag as much as you can, but not so deep in that you're losing your whole residue. And there's also been some question about whether it's possible for a pot to just soak in so much residue that, in essence, when we do a residue analysis, we're looking at the early use lifetime of the pot and not its later use. And I don't know that anyone's ever definitively come up with the answer to that. In theory, you would think that this would happen, but in reality, except possibly with something like an olive oil amphora or a jar full of petroleum tar, usually they don't seem to get so full of lipid that they just stop absorbing altogether.
Cool. Thank you very much, Dr. Reber. Okay, thank you everybody for coming today, and everyone here in the room and online. And I'd also like to welcome you all to our next lecture, which is going to be August 22nd, by Dr. Brooke Bauer, who's at USC Lancaster. She'll be talking about the history of Catawba Indian women during the colonial period. And so thanks very much for coming. If you want to look for our future events, you can check out the North Carolina Office of State Archaeology website, or like us on Facebook or Instagram. Thank you.
Thank you.